There is another approach to solve the multiple sequence alignment problem. In this approach, a strong pairwise alignment is taken as a block alignment, and in the block this alignment, step by step one another alignment is added and it generates a multiple sequence alignment. But in this procedure, the strong alignment, pairwise alignment is taken, and if there is a gap is generated in that alignment, it cannot be removed. And this alignment is based on this formula that once a gap is always a gap, because once a gap is created, it cannot be removed in this pairwise alignment, and whenever a new alignment sequence is added in the alignment, that gaps will remain as such. But in this approach, because the pairwise alignment that is taken as a source of the for multiple sequence alignment is very much similar, so that pairwise alignment added the most similar sequences to give up the multiple sequence alignment. So because the gap is created, so this alignment is reduced to k-1 sequences. So the motivation for the choice of these closest strings that are taken as a pairwise alignment, because whenever there is a closely related sequences for the pairwise alignment, the result will be more reliable. And we can see that the many of the softwares that are available and that are used for the multiple sequence alignment, they use this approach to calculate the multiple sequence alignment. So although progressive multiple alignment algorithms work well for very close sequences, so there are no performance guarantees for this approach. The problem with progressive multiple alignments algorithm like the program Cluster that use this approach to find out the multiple sequence alignment scores is that they may be misled by some spurious strong pairwise alignment in effect, that is a bad seed. And if very first two sequences picked for the building multiple alignments, here we can see this is the seed alignment, and if a gap is created in the seed alignment, here is the gap, so that the gap could not be removed up till the last step. So this is the main drawback of this approach. If there is a very first two sequences picked for building multiple alignments are aligned in a way that is incompatible with the optimal multiple alignment, the error in this initial pairwise alignment will propagate all the way through the whole multiple alignments. So many multiple alignments algorithms have been proposed, and even with systematic deficiencies such as the above, they remain quite useful in computational biology. And in this, the second example, we can see that this approach is useful when the similar sequences are taken for this approach. For example, if these first two sequences are used to generate the first seed pairwise alignment, we can see all of the nucleotides in these two sequences, they are aligned with each other. So whenever third or fourth or fifth sequence will be added, it will automatically align with these two already aligned nucleotides. So this approach is beneficial for the related sequences rather than sequences that create a gap. We have already seen that the multiple alignment for K sequences as a generalization of the pairwise alignment problem, which assume the existing k-dimensional scoring matrix, and we know that this scoring matrix is delta. Since such k-dimensional scoring matrices are not very practical, we have briefly described two other scoring approaches that are more biological relevant. The choice of the scoring function can drastically affect the quality of the resulting alignment, and no single scoring approach is perfect in all circumstances, as we have seen in the previous examples. So the columns of the multiple alignment of k sequences describe a path of edges in the k-dimensional version of the Manhattan added grid-like added graph. We have seen earlier that we can use that approach. Although it was a two-dimensional approach, we can use here a three-dimensional approach. The weights of these edges are determined by scoring functions, that is delta. So intuitively, we can assign higher score to the columns with a low variation in letters, such that high score correspond to highly conserved sequences. For example, the multiple longest common subsequence problem. The score of the column is set to 1, as we have seen earlier, as if all the characters in the column are same, and 0 if even one character disagree. We have seen that, for example, if this is the multiple sequence alignment, and we have seen that all these are A's is okay, but in this one there is a one gap and two T's. So the score will be here 0 and this score will be 1 and 1, because if we have 20 sequences and 19 are identical TTs, and if there is a one gap, the score will be 0. So for one score, all of the nucleotides at a particular point should be identical.